Before going over photosynthesis, it is important to discuss electromagnetic radiation. The sun emits an enormous amount of electromagnetic radiation in the form of gamma rays, X-rays, UV rays, infrared rays, microwaves, and radio waves. The most important type of electromagnetic radiation given off by the sun and used by plants is visible light, which happens to be the only electromagnetic radiation that is visible to the human eyes. The amount of energy found within a wave is measured by wavelength. Shorter wavelengths have more energy and are dangerous to living things. This is why gamma rays are really dangerous and X-rays and ultraviolet rays are harmful in certain levels of exposure and time. A wavelength is measured from crest to crest, crest to crest. The bigger the distance, the farther the waves can travel, but the less amount of energy is available within those waves. The closer they are together, the least amount of travel they can do, but they can contain more energy. So visible light kind of has that happy medium. Visible light is found between 400 and 700 nanometers, which is the distance between wavelengths, on the electromagnetic spectrum. We view light as white, but it is actually made of 7 colors: red, orange, yellow, green, blue, indigo, and violet. Violet runs closer to 400 nanometers and therefore has more energy in the wave, but it can't travel as far, whereas red runs closer to 700 nanometers, traveling further, but it has less energy in the wave. The colors we perceive are a result of pigments reflected or transmitted by the plant. For example, green light is not typically absorbed by plants and is therefore perceived as green by us. If you take a look at this chart, you'll notice that the absorption of light on the y-axis and the wavelength on the x-axis, green coloration does not have a lot of absorption, so it's reflected by most plants, which is why they appear green in color. These pigments and leaves are used to absorb different wavelengths of sunlight on the absorption spectrum, and they're found in 2 groups. First is chlorophylls. There are 4 types of chlorophyll that exist: A, B, C, and D. Since flowering plants are often used as a model to teach photosynthesis, we will just focus on chlorophylls A and B. These chlorophylls differ slightly in absorbing violet, blue, orange, and red light, and are more commonly used to carry out photosynthesis. Chlorophylls reflect or transmit green light. Transmission means the light simply travels through the leaf. Carotenoids, which include lycopene in tomatoes, zeaxanthin in corn kernels, and beta-carotene in carrots and other orange-yellow fruits and vegetables. These absorb more green and blue light while transmitting orange and yellow light. Carotenoids are also mixed with chlorophylls in green leaves. These are important as chlorophylls can be damaged by too much sunlight, whereas carotenoids can transmit excess sunlight as heat, preventing damage to plant leaves. Most plants contain a mixture of different pigments to help them maximize sunlight absorption throughout different times of the day, as well as being adapted to growing in well-lit or dim places. Now we will go over the details of photosynthesis. Just like in my cellular respiration video, this is designed for first-year science majors and AP biology classes, so the information may be more than what's necessary for a general biology class for non-science majors. Likewise, it may not have enough detail if you're studying plant physiology or biochemistry. Photosynthesis, again, can be broken down into 2 stages. The light-dependent reactions, which generate ATP and extracts hydrogen ions and electrons to load onto NADPH. And the Calvin cycle, which receives that ATP and the NADPH to combine carbon dioxide into the hydrogen ions and electrons delivered by NADPH to help make precursor molecules for glucose. The light-dependent reactions work within the membranes of the thylakoids with some activity inside the cytoplasm of the thylakoid. On the surface of the thylakoid membranes lie 2 types of photosystems, 1 and 2. Both of these are protein complexes embedded with pigment molecules. These were named from the order in which they were discovered, but the light reactions utilize photosystem 2 before photosystem 1. This is perhaps the most elaborate PowerPoint slide I have ever made, so I will take this one step at a time. Also, for the sake of simplicity, I'm leaving out some of the finer details, but it shouldn't affect the overall understanding of photosynthesis. First, step 1. Water enters the thylakoid, oxygen is removed, and the hydrogen ions create a chemiosmotic gradient inside the thylakoid space. The electrons lead to the reaction center of photosystem 2. Next, sunlight is then received by the pigment molecules of photosystem 2, which transmits a photon, which is that tiny yellow dot you saw. A photon is a packet of light energy that, when transmitted between pigment molecules, helps to increase the potential energy as it excites electrons within photosystem 2. Eventually, it reaches the reaction center where the electrons from water are excited or gain more potential energy. Notice the 3 large protein complexes in the middle of the membrane. This is the photosynthesis electron transport chain. These 3 proteins move electrons from photosystem 2 to photosystem 1. As this happens, hydrogen ions are able to be pumped into the thylakoid, later to be used by ATP synthase. Unfortunately, these electrons lose some energy from photosystem 2 because of this, and then they enter the reaction center of photosystem 1. So more sunlight hits photosystem 1, producing another photon to excite the electrons within the photosystem, providing these same electrons with more potential energy. Afterwards, the electrons are collected by a protein called ferredoxin, which then delivers the electrons to NADP+ reductase. This enzyme converts NADP+ into NADPH, which now houses a hydrogen ion and 2 electrons. Now, the hydrogen ion hasn't made it yet, because I need to show you what happens to the hydrogen ions inside of the thylakoids. Just like NADH of cellular respiration, NADPH is an electron carrier used to collect hydrogen ions and electrons from the light reactions to be used by the Calvin cycle to help build precursor molecules for the glucose. As the electrons are loaded into NADPH, hydrogen ions pass through ATP synthase to force a bond between ADP and phosphate to make ATP. A hydrogen ion is then going to be bonded to NADP+ and the 2 electrons to make NADPH. ATP production within the chloroplast is self-sustaining, meaning that the ATP is used to power the Calvin cycle, which we'll discuss next. The rest of the plant cell relies on the mitochondrion to produce ATP to carry out its other cellular functions. The light reactions take place in the thylakoid membranes of the plant's chloroplast. So let's take a look at the inputs and the outputs of the light reactions. First, you need a water molecule, which comes from the roots or water vapor through the stomata. You need 6 NADP+ and 9 ADPs and inorganic phosphates, which both come from the Calvin cycle. When the light reactions are over, oxygen is then going to leave the system. 10% of that will be used to help make ATP in the mitochondrion, but 90% of it goes out into the atmosphere, which is, necessary for animals like us. 6 NADPHs and 9 ATPs are then sent to the Calvin cycle. After ATP and NADPH are produced by the light reactions, they'll go to the Calvin cycle where carbon dioxide is collected. This carbon dioxide is combined with the hydrogen ions and electrons from NADPH and is powered by ATP to make the precursor molecule for plant sugars, G3P. In simplicity, glucose is an end product of photosynthesis, but so is fructose, sucrose, and starches. The Calvin cycle takes place in the stroma, or fluid-filled spaces, of the chloroplast. The Calvin cycle is broken down into 3 steps. Just like the citric acid cycle in cellular respiration, this is a repetitive chemical reaction. Before we begin, let's look at the inputs of the Calvin cycle and their origins. Carbon dioxide comes from the atmosphere through the stomata of the leaves. NADPH and ATP both come from the light reactions we just discussed previously. The first step is called carbon fixation. as the carbon source for plant sugars comes from atmospheric CO2 that enters plant leaves through their stomata. In order to do this, we need 3 ribulose biphosphate molecules in the presence of a very important enzyme called ribisco enzymes. These 3 CO2 molecules end up making 6 3-phosphoglycerates. The 3 means that the third carbon contains a phosphate group, and this is carbon fixation. In step 2, we now need to reduce or add electrons and hydrogen ions to the 3 phosphoglycerates. To do this, we need 6 ATPs, which help power the enzymes to help with this process. And please remember that the enzymes are not shown, except for ribisco, but they are there doing the work in the background. We also need 6 NADPH molecules to deliver these hydrogen ions and electrons necessary to reduce the 3 phosphoglycerates. The NADPH molecules transform to NADP+, and the hydrogen ions and electrons are transferred to the 3 phosphoglycerates to reduce these molecules into 6 molecules of glyceraldehyde 3-phosphates, which we abbreviate as G3P. Only one of these is actually used to make glucose, fructose, sucrose, or starches later on. But since this is a 3-carbon molecule, you will need another 3-carbon molecule of G3P to help make glucose or fructose, since both of these are 6-carbon molecules. The remaining 5 G3P molecules will be used in the next step. So the last step of the Calvin cycle is the regeneration of ribulose biphosphate so the cycle can start over. 5 G3P molecules will combine with 3 ATP molecules to help build 3 ribulose biphosphate molecules. This allows the Calvin cycle to begin again with more carbon dioxide. The outputs for the Calvin cycle are 1 G3P, which is made into sugars, 6 NADP+, 9 ADP plus inorganic phosphates, which are both sent to the light reactions to receive more hydrogen ions, electrons, and to be converted into ATP. Photosynthesis and cellular respiration are 2 of life's most crucial chemical reactions. The products of photosynthesis become the reactants of cellular respiration, and the products of cellular respiration become the reactants of photosynthesis. So it's a quite cyclical chemical reaction that all life on Earth depends upon. I like this picture, but let's not forget that plants perform both reactions. Also, most oxygen on this planet is actually made from algae in the ocean, since oceans take up 75% of the Earth's surface. It's ultimately important to protect nature so that it continues to allow us to function as living things. Although we have spent the past 2 chapters going over some intense biochemical reactions, I hope you can step back and appreciate this wonderful process of life, that to be alive requires energy, and that energy ultimately starts with the sun. In our next chapter, we will see how cells make more of themselves in the processes of mitosis and binary fission.